Hello all. Today I will talk about the agarose gel electrophoresis, which is used in DNA separation in molecular biology lab. Here is the device. It consists of the power supply, which is used here in this shape, in this picture. Here is the tray inside the tank, the huge tank here, and there are a tray inside the tank. Which is agarose. Agarose is a polyacrylamide. It's a polymer of carbohydrate. It's extracted from seaweeds, and gel electrophoresis technique is a method that uses an electrical current and a gel matrix to separate molecules like DNA and proteins. Buffer, a solution containing either a weak acid and its salt, or a weak base and its salt, which is resistant to change in pH. And in gel electrophoresis, we use a dye, which is a fluorescent chemical that uses to demonstrate or to visualize the DNA. Its name is ethidium bromide. It's a fluorescent chemical that intercalate between the bases base pair in a double-stranded DNA molecule. Here we shall focus on this. It needs double-strand DNA in order to be intercalated inside. Commonly used to detect DNA following gel electrophoresis. DNA ladder consists of known DNA size used to determine the size of unknown DNA sample, and the DNA ladder usually contain regularly spaced size sample, which when run on an agarose gel looks like a ladder. So the name DNA ladder came from the shape of the fragments of DNA that demonstrated in the picture used for DNA. Power supply, a source of electric current. Then the agarose is a polyacrylamide obtained from agar and consisting of linear polymer repeating unit of D-glycosylgalactose and 3 to 6 anhydro levotertiary galactose. It's extracted from seaweed and purified for use in electrophoresis. This is a microscope, electron microscope for the agarose gel, and this is a powder that used to make the gel. The movement molecule through an agarose gel is dependent on many parameters. One of them is the size of the molecule and the charge of molecule, the pores of the agarose gel, and the electrophoresis buffer used is concentration of the buffer. Firstly, we have to talk about the size. The size of molecule that used in this experiment is edges. It's edges cutting per meter in this experiment, because if the size is big, it faces hindrance from the pores of agarose gel, so its movement is slow. Vice versa, the size of molecule that very small, it's migrate faster than that greater than the smallest one. Charge of molecule. The charge of molecule of DNA and RNA are negative, and it's a huge variation for proteins. The pore size present in agarose gel is dependent on the concentration of agarose, and the electrophoresis buffers, its concentration is one of the parameters. If the electrophoresis buffer is not concentrated, it's diluted, so the movement of the DNA is faster, and if the buffer is concentrated, then the movement is slow. Electrophoresis buffer used in this experiment is two types. One of them is trace acetate editor, and the other is trace borate editor. Trace acetate editor is favorable for proteins, and trace borate editor is favorable for DNA and RNA. There are some advantages of trace borate editor over using this trace acetate editor. One of them is the trace borate editor cannot make huge heat. The important parameter of the electrophoresis buffer are its composition, its ionic strength. The electrical conductance of the gel is dependent on the presence of ions in the buffer. Without presence of the buffer, the current running through the gel would be very small, and molecule would migrate either very slowly or not at all, because the conductance of the buffer is one of the parameters that make the experiment successful. Conversely, a buffer with too high an ionic strength produce a very high electrical conductance and significant amount of heat that make the experiment is not successful. The heat that is produced by passing the electrical current through the gel can be hot enough to denature the DNA, and the denatured DNA cannot visualize by acidium bromide, in order that the acidium bromide need double strand to be intercalated in it. Then the ethidium bromide is most commonly used nucleic acid stain agarose electrophoresis is intercalate double-strand DNA and RNA. When it is exposed to ultraviolet light ethidium bromide fluorescence, here as we see in here. Here in this lab we have two mutagen. The mutagen compound is the material that causes the cancer. There are two mutagen in this lab, the ethidium bromide and ultraviolet. Loading buffer, it's contain glycerol as density agent for the DNA sample, and tracking dye that consists of one of these material, the bromophenol blue, dienol xylose cyanol orange G. It doesn't matter if it contains 3 or 2 of this matter. Here is the buffer that uses the 50x TAE buffer, trace acetate editor, and its content, the preparation of it, and 10x TBE buffer recipe. Here is the material used in this experiment. Here is the devices. Here is the tank, then the power supply, and the wheels or electrical leads here. And this is a compass and casting tray. The procedure is measuring 1 gram agarose powder and add it to 500 ml flask, then add 100 ml TAE buffer 1x or TBE buffer to the flask, then melt the agarose in microwave until solution become clear. Then here this is the solution before the melting, and here is the solution after melting by microwave. Let the solution cool to 50 to 55 centigrade, in order to be suitable for adding the sodium bromide. So here is an point for here to mention is if we add the sodium bromide in heated solution here, the evaporation of sodium bromide can cause pneumonia and cancer, lung cancer for the user of this experiment. So it's very safe to use sodium bromide in low temperature, about 50 to 55 centigrade. And unfortunately, sodium bromide is heat labile or heat sensitive, so the high temperature can damage the sodium bromide, so it's not useful for this experiment if added in heated solution. Here is the measuring of the weighting of the powder of agarose gel, and here is before adding the 100 TBE buffer to it and insert it to the microwave. Then we have to seal the casting tray and put in inside the wheels here the comps. Then we have to put to lay the agarose gel solution after adding an sodium bromide to it and mixing it to it. Then we have to load the DNA. Before loading the DNA to the agarose gel, it must be mixed with loading buffer, the loading dye or loading buffer. The loading dye concentration is 6x, and the solution must be in ratio of 1 to 6 of sample, 1 of the loading dye to 5 of sample. Here is the running of the sample. It's run to the red. We have to repeat it. The run is to red from the cathode to anode. Here is not the result of DNA. It's the running of the DNA. It's about 30 minutes. This picture is shown here. The purple color is for the lincyanool, and the sky blue color is for promphenol blue, and there are no visualization of DNA exactly here. It's only the loading dyes shown here. Here is the DNA bands, which is visualized by the UV light. Again, in these pictures, we can see the sequence of steps that used. They prepare the agarose and they mix it with the loading dye. Then they load it to the tray. Then they apply the power supply, the electricity in it. Then it's put it in the UV light or documentation system. Then this is the picture that you picked up by the gel documentation system. Here is an ideal result for DNA electrophoresis. Here is a comparable result. Here is the same loading DNA, but the agarose concentration is different. In this picture, there are a naked circle, which is or linear or supercoiled. Here is an ideal result of DNA electrophoresis. Also, they are mentioned in this PowerPoint a lot of problem and its solution. Any question should be commented in the video, and goodbye, and see you in the next video.